Hello everyone, my name is KAUSALYAH SELVARAJU and my matrix number is SB20006, and today I want to present about the synthetic biology about Artemisinin. And now I would like to start my presentation with the introduction. It is the branch of research that entails giving organisms new skills so they can be redesigned for practical reasons, and also assembling a biosynthetic pathway of genes from a plant, Artemisinin annua, and other organisms into bacteria using synthetic biology. One of the first success stories in synthetic biology is engineering biology to generate a drug on an industrial scale, and was the synthesis of semi-synthetic Artemisinin. And the WHO approved the semi-synthetic Artemisinin for use in the production of antimycin derivatives for inclusive in antimycin combination therapies after it was demonstrated to be functionally equal to the medicine generated from plants. Malaria is a global health problem, so that synthetic biology had made research using some herbs and plants to produce artemisinin, which can prevent malaria. Multi-drug resistant strain of the malaria parasite, Plasmodium falciparum artemisinin, a complex chemical extracted from the plant Artemisia, is highly effective. This flowchart shows the artemisinin biosynthesis pathway occurs in the glandular trichomes of Artemisia annua, which produces the artemisinin. Let's continue with how this research went to synthetic biology to produce a product. The production of semi-synthetic Artemisinin was one of the first synthetic biology success stories. Engineering biology to produce a pharmaceutical at industrial scale. Malaria drugs have contributed significantly to the reductions of malaria mortality and morbidity. The focus for many years has been to screen traditional medicine to find new anti-malaria drugs. The malaria drug Artemisinin is an example of this and originated from A. annua, a Chinese medicinal plant commonly known as sweet wormwood. It was discovered by the Chinese researcher Yu To and her team in 1972, and it was named Qinghaosu. And then chemically, Artemisinin is a sesquiterpene lactone with a unique endoparasite structure without the nitrogen containing heterocyclic ring like other anti-malaria compounds.
Synthetic biology regarding Artemisinin technology is not available in Malaysia. However, there is a research from University Malaya regarding the enhancement of Artemisinin production with UVB radiation and dimethyl sulfoxide in Artemisia annua L cell suspension cultures, and also a clinical trial of Plasmodium falciparum Artemisinin resistant. It has been proven that the endoparasite sesquiterpene lactone Artemisinin, which was isolated from the Chinese plant Artemisia annua L, exhibits a variety of unique biological actions. Artemisinin and its derivatives have shown anti-parasitic, antiviral, antibacterial, anti-cancer, and anti-genetic effects through a variety of modes of action. However, there is a possibility of significant shortage in Artemisinin availability because of the low accumulation of Artemisinin content in planta. Despite extensive effort to tackle this issue, the commercial manufacture of this helpful medicine is hampered by a lack of economically viable protocol. Consequently, using annual cell suspension culture to make more Artemisinin may be a good way to solve the problem of not having enough Artemisinin by boosting. In this study, UVB and dimethyl sulfoxide were chosen as inducers to see how they affect Artemisinin production, with a focus on the last non-enzymatic photooxidation composition step that leads to the formulation of Artemisinin. There is a clinical trial about spreading Plasmodium falciparum Artemisinin drug-resistant threatens global malaria public health gains. Limited data exist to define the extent of the P. falciparum Artemisinin resistance of the Greater Mekong region in Malaysia. Patients with uncomplicated falciparum malaria and a parasite count below 100,000 per liter were admitted to treated by this hospital in Sabah, Malaysia. For a clinically effective study of oral artemisinin, total could lose 12 mg per kg daily for 3 days. All of the patients got 25 mg per kg of mefloquine in 2 doses on day 3 and 4, for a total of 25 mg per kg. They were on watch for 28 days. Also, between 2012 and 2016, 21 KAUSALYAH polymorphism linked to artemisinin resistance in P. falciparum were looked at in P. falciparum isolated from people who went to hospital, mostly the tertiary referral area of Western Sabah. As a result, in all, 49 people were signed up and given oral artemisinin. By 48 hours, 44 of the patients had been cured of parasitemia, and by 72 hours, 49 had been cured. The geometric mean number of parasite representation was 95%, and it took 4.3 hours for 50% of the parasites to leave the body. 3 of the patients had a parasite clearance slope half-life of less than 5 hours. For KAUSALYAH markers, all 272 P. falciparum isolate that were looked at were wild type. In this pre-elimination setting in Sabah, there is no evidence of endemic P. falciparum that is resistant to artemisinin. For uncomplicated malaria in Sabah, the current treatment recommendation, which call for ACT as the first line of treatment, are still valid. In the southern east of the Greater Mekong subregion, continuous observation is required.
For large populations around the world, including those in Africa, Brazil, India, and many other places, malaria remains a serious threat. It is one of the more worrying local problems in Malaysia, along with the increase in malaria cases. Synthetic biology technology can be applied in a variety of ways to tackle the local issues of rising malaria cases. In general, this is how artemisinin is extracted directly from plants. The desired plant, Artemisia annua, produces artemisinin through the trichomes of its leaves. The resulting artemisinin is then obtained through extraction and purification. Later, the chemical conversion is done to convert the artemisinin into its other derivatives. Using synthetic biology, researchers have found new ways to enhance the production of artemisinin. By introducing new genes and engineering a different metabolic pathway in Saccharomyces cerevisiae or Escherichia coli, scientists like Dr. J. Kiesling are able to synthesize microbial-based artemisinin by letting those microbes undergo fermentation. Genes from the plant Artemisia annua, which are responsible for producing artemisinic acid, are introduced into the desired microorganisms, and now these microbes are cultivated to undergo fermentation to produce artemisinic acid. This process is called the semi-synthetic method to produce artemisinin. In 2006, the team described how they had genetically modified a strain of yeast called Saccharomyces cerevisiae to be able to produce large amounts of artemisinic acid, which is the direct ancestor of artemisinin. Dr. J. Kiesling and his team engineered the yeast in a 3-step process, first introducing bacterial and Artemisia annua plant genes into the yeast's DNA that interacted with the yeast's own genes to produce amorphidine, a chemical precursor to artemisinic acid. This new metabolic pathway was similar to the one created in E. coli. In order to produce the enzyme P450 that the plant uses to transform amorphidine into artemisinic acid, they finally cloned the gene from Artemisia annua and expressed it in the yeast strain that produces amorphidine. In this situation, artemisinic acid is toxic to cells and will be excreted as a waste product, eventually clumping up as solid white particles. This method of producing artemisinin is the most efficient and pure way to produce drugs because the desired drug is produced outside of the cells and remain in the bioreactor. This allows for the drug's purification at the end of the fermentation without the use of an expensive product extraction method. When the yeast discovery was first made, it was noted that microbial production of artemisinic acid is a viable source of this powerful family of anti-malarial drug due to the existence of known relatively high-yielding chemistry for converting it to artemisinin or any other desired derivative. Instead of being marketed as a monotherapy, microbial-based artemisinin can be bundled as a part of an anti-malarial drug cocktail to delay or even prevent the emergence of resistant malaria parasites. Another way that synthetic biology opens the door to treating malaria is through gene therapies that targeted the malaria-causing parasite. Through gene therapies, it is possible to change the parasite's genome and prevent it from replicating. It can be used to weaken the parasite before it contacts a human host, and this is a crucial step in fighting malaria. Gene therapies offer a more effective method of treating malaria because they directly target the parasite. Overall, synthetic biology is an effective weapon in the fight against malaria. Although the use of synthetic biology in the fight against malaria is a promising prospect, it raises ethical and health issues that must be considered when utilizing this technology. These issues include the potential of creating a new unnatural form of drug which could lead to unknown side effects. For example, there is a fear that synthetic biology may lead to the emergence of new strains of malaria that are resistant to the existing therapies. As evidence, consider that recent studies have already suggested that mutations in malaria-causing Plasmodium parasites have already developed, leading to a new form of drug-resistant malaria in the Malaysia-Thailand border region. This is an extremely concerning development, as the emergence of drug-resistant malaria strains could potentially cause a public health crisis. Moreover, another ethical dilemma is the potential for companies to exploit the developing world by charging high prices for synthetic biology-based treatments. This could limit access to essential drugs and services in countries with fewer resources, where people are already struggling financially. Additionally, there is a concern that this technology could be misused to create a new form of the drug that could be weaponized, creating an ethical dilemma in terms of implications of allowing access to such a drug. These ethical dilemmas need to be addressed to ensure that synthetic biology is used safely and responsibly in future.